Hi, my name is Brian Kirz. I'm a first-year PhD student in Nancy Chen's lab at the University of Rochester. And today I'm very excited to share with you in my first talk the novel draft assembly for the North American red squirrel, also known as Tamaeoceras hudsonicus, and discuss a little bit about our investigation into why its genome size is so large. So to start, genome size is a fundamental biological trait, but despite that, we don't really understand the evolutionary causes, mechanisms, and consequences behind it. So for example, what kinds of selective pressures are regulating the accumulation and loss of DNA? Why do some groups, like mammals, show comparatively little variation while other groups show a lot? We have ideas, but these are still really open questions, and if we look at outliers, across small phylogenetic distances, we may be able to shed some light on the answers to some of these evolutionary questions surrounding genome size variation. And today I'd like to share with you one example of that. So we did not expect to find this when we set out to assemble the genome of the North American red squirrel, but we've discovered that it has a much larger genome size than other related squirrels. So on the left, you can see a simplified phylogeny of the family Sirinae. That is the tree squirrels combined with the flying squirrels. I've selected the species here to display based on phylogenetic proximity, as well as which ones have published assemblies that we can then use to analyze. I've also included divergence times, very rough, for the species here. As you can see, Tamaeoceras hudsonicus diverged from its closest ancestors, closest relatives in this tree about 17 million years ago, which for context is roughly the same distance between humans and orangutans. On the right, you can also see the comparison of genome size estimates across 3 of these species. Since we don't have C values or flow cytometry data for any of these species except for the gray squirrel, I decided to use K-mer-based whole genome size estimation analysis in order to do this. Basically what that does is it's looking at the distribution of identical sequences of a certain length over a bunch of short reads. There aren't published random shotgun data for every single one of these species, so as you can see, 2 of them are missing, but it still shows that, that T. hudsonicus has the largest genome size. I wanted to show this figure anyway because it's a separate methodology, different from assembly size, that can also reinforce some of the conclusions that we can draw from it. We do expect that K-mer size estimation is going to be a little bit different from assembly size. Some of the assumptions that underlie the methodology are different and can break down when you're dealing with genomes that have a lot of repetitive content, and I'll get back to that later. So here's the comparison of assembly sizes across the family Sirinae. As you can see, T. hudsonicus has the largest by far at 6.1 gigabases, and the rest of them are clustering very tightly in this sort of 2.5 to 3 gigabase range. If I were to zoom out on this phylogeny and show you the assembly sizes for all of the squirrels, including groundhogs, chipmunks, that kind of thing, you would still see this pattern. So a very, very tight band around 2.5 to 3 gigabases. I'd also like to emphasize that all of these assemblies are very high quality. They've been published in the last 5 years. They're all long read data, and some of them have high C data too. So this is not an artifact of methodological differences. We think this is real. So how could this expansion have occurred? I'm going to break it down into 2 sort of very broad categories, the first of them being genome duplication. This can be as large and as drastic as a whole genome duplication event or something a little bit more minor. Given that there's so much coding genetic material involved, often this type of thing has major phenotypic consequences, meiotic disruption, gene dosage problems, cytoplasmic incompatibility. And so you may expect to see larger phenotypic changes. Plants get away with these kinds of extreme polyploidy all the time, but it's much rarer in animals and almost unheard of in mammals. So the karyotypic, visual karyotypic data that we have for T. hudsonicus from the 70s shows that it's not polyploid, it's definitely diploid, and it has 23 chromosome pairs, which is well within the ballpark of its cousins, the closest of which have 20. The other potential cause of this expansion in genome size is repetitive element amplification. So there are many, many ways to classify repetitive elements, but I'd like to break them down into two. The first being tandem repeat sequences. These are things like satellite DNA, short, simple interspersed sequences that typically proliferate passively, either through slippage during DNA replication or unequal crossing over during recombination. The other group are transposable elements, or TEs, which proliferate themselves actively, either by themselves or leveraging the machinery of another TE to do the work for them. In small quantities, this is relatively harmless. In fact, it's actually quite common in mammals. But left unchecked, TEs can get into places where we don't want them. So interrupting functional genes, messing with expression, that kind of thing. Now, the strategies that genomes have come up with to regulate this TE overrun are not fully understood, but at least for in relation to genome size variation, in birds and mammals, it's been proposed. There's been proposed sort of an accordion model whereby over generations you get a gradual accumulation of repetitive content, which is then kept in check by large deletions, and that's why we see the sort of similarities in genome sizes that we do. So T. hudsonicus is not the first mammal or even the first rodent to have been shown to undergo a rapid genome size expansion in a relatively short period of evolutionary time. This is the Vizcacha rat of Argentina. It is has the largest mammalian genome that we know of at 8.4 gigabases, which makes it about twice as large as its closest relative, from which it diverged about 5 million years ago. And a 2017 analysis of WGS data concluded that there was not much support for a whole genome duplication event, and that the genome expansion was primarily due to a rapid expansion in repetitive content. So in light of all this, in light of the fact that these whole genome duplication events are pretty rare in mammals, we're not expecting to find them, the Tamiaceras hudsonicus is phenotypically very usual or typical of squirrels, even though it has an unusually large genome. And the precedence of this Vizcacha rat case, we hypothesized that the genome size expansion was primarily due to this repetitive element amplification. So in order to test this, I gave myself two tasks. One of them would be to assemble a high-quality genome assembly for Tamiaceras hudsonicus using long reads and high-C data, the best we could get. And number two would be to test the mechanisms that might underlie that genome expansion with a particular emphasis on our hypothesis of repetitive content. So here's the workflow, the methodology that I used to assemble the genome. We combined PacBio Hi-Fi and Oxford Nanopore long reads together. We used high-C contact data in order to scaffold that and make it better. And then we evaluated genome completeness with BUSCO and assembly statistics with QUAST, as well as comparing them to other published squirrel assemblies. I am very happy to say that after all is said and done, the Tamiaceras hudsonicus genome has the lowest number of scaffolds and the highest mammalian gene completeness out of any of the dozen or so published squirrel genomes, so we're very satisfied with the quality. And this assembly would then be used as the basis for our repetitive analysis of the genome, which I'm about to show you, where we break it down by repeat family. So this is the proportion of the T. hudsonicus genome that is taken up by repetitive content. As you can see, it's a lot, almost 2/3. In context, remembering that the genome size is about 6.1 gigabases, we're looking at 3.8 gigabases of just repetitive content. Remember, the eastern gray squirrel has the entire genome size is 2.8 gigabases, and this is actually larger than that. I'd also like to point out in this photo, in this figure, that the largest classified repeat families that we see, lines, LTRs, signs, and DNA transposons, these are all TEs. We're not sure which of them are active or not, but that's something to keep in mind going forward. When you put the T. hudsonicus genome assembly at repeat content compared to its squirrel relatives, you see that there's a massive increase in repetitive content across families. So it's not just concentrated in one family. We see this expansion across LTRs and DNA transposons and so on. one of the things that I think this figure doesn't do justice is that because we're adjusting from genome for genome size, the actual absolute increases in this repeat content is larger than it even looks in this figure. Because if I were to extend the y-axis to capture the true number of base pairs that we're seeing here, it would be much larger, given that all of these other squirrels have assemblies that are about half the size. So to review the project, we first developed a pretty high-quality genome assembly for T. hudsonicus, leveraging long-read and high-C data. Our results support that there is a real genome size expansion, and that has been primarily due to the amplification of repetitive content, at least, which is not exclusive from our other hypothesis. I'd like to emphasize that this is a first year's work in progress, so going forward, I'd like to make the genome better. I'm already doing that by trying to get to a chromosome level assembly using our high-C data. One of the questions that's going to be able to let me answer is understanding more about the distribution of repetitive content within the genome. Where is it? On which chromosomes? How old are these expansions? Is this ongoing? Did this happen in a short burst or gradually? One thing that will make that easier is the standardization and curation of the repeat libraries that I use for analysis. So combining all the de novo stuff that I've done with Repeat Modeler on my assemblies and taking together that, what's publicly available on RepBase and DFAM, putting it all together and standardizing that methodology. Another way we can improve the genome is to annotate it. We already have RNA-seq data. That's up next. And then this will allow us to quantify and investigate that alternate hypothesis for genome expansion, which is the duplication of genetic content. I would like to thank the Chen Lab at the University of Rochester very much. It's a wonderful place to work. I'd like to thank my funding sources, my great collaborators, and if you have the chance, please come visit my lab mates' talks tomorrow. I'd like to thank all of you for being here, listening to me, and giving me an opportunity to share my work. And with the remaining time, I'd be happy to take questions. Yes? Yeah, so that would be up next. We decided to push that part back because I've sort of looked down the rabbit hole of repeat elements, and I realized I could probably spend my entire career there. But that's very interesting to me, and I would like to know which repeat elements are still active and sort of the timeline of this expansion. Yes. So I would really love to get some more samples from different areas. This is a pretty widely spread species across North America. I don't know if there's a fluctuating environment or small bottleneck that's responsible for this kind of that's responsible for this genome expansion. I'm surprised that it hasn't. We don't see it as often in other things if it's due to that. But that's definitely one of the things that I would have to look into. I just need a better sort of understanding of what the natural histories of all of the different squirrels are in order to make that comparison. Thank you very much.
Well, thank you all so much for coming today. I'm going to be talking about the process of assembling the Anolis grami genome and then how we've used this genome to answer a couple of different evolutionary questions. So first, I want to introduce my study system, Anolis lizards. So anoles are a genus of lizards that's found throughout the West Indies and in Central and South America. And we currently have over 400 recognized species in this genus, and they exhibit a wide range of diversity. Now, anoles have also been used as study animals for diverse areas of research, spanning community ecology, communication, behavior, adaptive radiation, you name it. I feel like anoles have been studied for that. But unfortunately, we only have a few genomes for this diverse group of lizards. And although we have several topics in evolutionary biology that have been elucidated by the study of anoles, progress in some areas has been hampered by limited genetic information in this group. Now, one of the species lacking a genome assembly is Anolis grami. This is the Jamaican turquoise anole. So they are native to Jamaica, and they have been recently intentionally introduced to Bermuda as a pest control mechanism. because we know that's a good idea and it always works. We currently have 2 recognized subspecies in this genus. So we have grami here on the left. This is found throughout most of the island. And then this smaller form, grami aquarum, on the right, which is the eastern form. So it's restricted to areas east of the Blue Mountains. Now, they're trunk crown anoles, so they're found from around eye level on the tree up into the canopy. And a little fun fact is that they're one of the few anoles that vocalize. So Anolis grami is one of 7 species within the monophyletic radiation of Jamaican anoles, but we still have no reference genome for this group. So a complete and contiguous reference genome is needed to answer some of the interesting questions that we may have, specifically questions that are brought up from past research in Jamaican anoles and grami in particular. So work by Todd Jackman on the phylogeography of this species has shown that different geographic populations of Anolis grami are highly genetically divergent, so this suggests that maybe this is a complex of species. And then also there's been some really interesting chromosomal evolutionary patterns in this species that I'll talk about a little bit later. So then I just want to briefly talk about our genome assembly. So we currently have 10 published anole genome assemblies, and I've labeled grami here with an arrow, so we have comparable completeness and contiguity. So contiguity is labeled here on the x-axis, which just measures how broken up our genome assembly is. So a higher contiguity means less broken up, and we're approaching chromosome scale ideally at the higher end of that scale. And so we have pretty good contiguity here. And then we have completeness measured on the y-axis as the percentage of expected vertebrate genes that we are finding in our assembly. And so we have pretty good completeness too. Now, along with assembling this genome, I have also generated an annotation, and that has pretty good completeness scores as well. So now that we have this high-quality genome assembly, we can use it to answer different evolutionary questions, and so I'm just going to talk about a couple today here. The first one being, how have sex chromosomes evolved in this lineage? And then secondly, where is the Jamaican clade in the anole phylogeny? So first, thinking about sex chromosome evolution in this lineage. So we know that sex chromosomes are incredibly important, and they play an outsized role in evolution, impacting processes like adaptation and speciation. But most of our knowledge of sex chromosome evolution comes from detailed investigations in a handful of representative lineages. But studying sex chromosome evolution in an adaptive radiation would allow us to examine their role in adaptation and sympatric speciation. Now, anoles are a perfect study system for this work for a few reasons. They're well known for their replicated adaptive radiations in the Greater Antillean Islands. They also share a single sex determining mechanism, so they have genetic sex determination. But we do see widespread chromosomal variation in these lineages, specifically within the Norops subgenus of Anolis. And so we have examples of lineages exhibiting homomorphic sex chromosomes, heteromorphic, we have XY, XXY, specifically within Norops as well. Now, one lineage that has shown some really interesting chromosomal evolutionary patterns is Anolis grami. So in 1986, Blake sampled different grami populations from across the island of Jamaica and found within population variation in the total chromosome number. And then this was echoed by Gamble et al. In 2014 that also showed this intraspecific variation in total chromosome number. So we have an estimated 2N from around 29 to 37. Now, furthermore, anoles in the Norops subgenus, from which grami is included in, have experienced widespread sex chromosome evolution relative to the hypothesized ancestral X chromosome for anoles. So using our genome assembly in combination with population genetic data from 50 males and 50 females, we've determined that scaffolds 11 and 12 correspond to the X chromosome in this species. So anoles are an XY system, and our genome animal is a female, so we're not able to actually pick up the Y chromosome in our assembly, but we can pick up regions that are indicative of the X chromosome. So we've used a few different lines of evidence to determine the sex-determining regions in this species. So first, we performed a genome-wide association study looking for male-associated SNPs. So these results are shown here in red and blue, with the red corresponding to scaffold 11, and then red and yellow, the yellow corresponding to scaffold 12. And so we see this very obvious signal here on the first half of scaffold 11. And this is indicative of alleles that are going to be associated with maleness. So these will be found on the non-recombining region of the Y chromosome, but are not diverged enough, so they are still aligning to the copy on our X chromosome here. Now, second, we performed a relative read depth analysis. So this is this black line here on our plot. This is showing the male to female read depth ratio. And then we see here on the last half of scaffold 12, this dip in relative read depth where males are having approximately half of the read depth that females do. And so this would indicate regions on the X chromosome that are no longer present on the Y, likely due to some degree of degradation. So because females have 2 X chromosomes, they will have double the read depth that males will. And then lastly, we've looked at the location of Anolis carolinensis X-linked genes. And so this is shown in purple here at the top of this graph. And so we see that these regions line up exactly with this last half of scaffold 12 that indicates the non-recombining region of the X chromosome. So through these 3 lines of evidence, we conclude that scaffolds 11 and 12 make up the X chromosome in this species. And I also want to say that we believe these scaffolds comprise 1 chromosome, and we're not picking up 2 different X chromosomes. So from our high-C data, you can do interaction mapping, and we see a high degree of interaction between scaffolds 11 and 12, which would indicate that they are 1 chromosome. So now that we have identified the scaffolds corresponding to the sex chromosome in this species, we can investigate sex chromosome evolution in this lineage. So Anolis carolinensis is hypothesized to have retained the ancestral X chromosome of Anoles, so it's conserved in this species. Unfortunately, I don't have a synteny plot of carolinensis and Grami to show you here today, but we do find that the Grami X chromosome is the result of a fusion between the ancient X and a couple of the carolinensis autosomes. We can look also at clade differences within Norops. So both Segrii and Grami are within this subgenus Norops. And so this is the synteny results between Anolis Grami, here scaffolds 11 and 12 on the bottom, and Anolis Segrii. And so we do see these clade differences. So the Grami X chromosome is syntenic with the Segrii X, which is chromosome 7 here, as well as a couple different autosomes 9 and 11. And so we're seeing these different evolutionary patterns within the Norops lineages, which is not surprising as previous work has shown this as well, this extensive sex chromosomal variation within Norops. So that's just one insight that we have gleaned from this genome assembly. I'm going to switch gears a little bit here and answer another question that we had is, where do Jamaican anoles fall in the Anolidae phylogeny? So we have conducted a large-scale phylogenomic analysis of all published high-quality squamate genomes to try and clear up some of the relationships within squamata being snakes and lizards. So in this analysis, we have 116 squamate species with 3 reptile outgroups, and we use the vertebrate data set, the Busco data set of genes for this, which total to 3,201 genes going into this analysis. So our tree provides some insights into a few different areas of the squamate phylogeny, like pleurodont iguanian relationships, as well as toxicopheran relationships. But as other recent squamate phylogenomic analyses have recovered similar results, I'm just going to focus on one insight, which is the placement of Jamaican anoles. As no other phylogenomic analysis has included a Jamaican species, this is the first opportunity to evaluate their placement using genomic scale data. So Jamaican anoles are within this Norops subgenus. So these are the Norops lineages. We have anoles in Cuba, their range shown in this maroon color here. We have our Jamaican anoles shown in green, and then South American Norops anoles, this range shown in blue. South American Norops anoles are sometimes found to be monophyletic, sometimes not, but when they are found to be monophyletic, we call them this clade Draconura. And so I'll be referring to them as that. So we have a couple different hypotheses for the placement of Jamaica within the family Anolidae. So phylogenetic studies in anoles using different data sets have given us these different hypotheses. So first, here on the left, we're finding a monophyletic Draconura and then Jamaican anoles as sister to this group to the exclusion of Cuban anoles. Or studies have found Jamaican anoles as nested within South American anoles. So rendering Draconura paraphyletic, and we have these two different South American anole clades coming up. So this auratus clade and the apletophalus clade. And so now we can investigate these alternatives using genomic data, as we have the first Jamaican anole genome assembly, but we also have genome assemblies from these different South American clades. So we have a genome for auratus and for apletophalus. So this is a zoomed in view of this larger squamate tree that I've shown you, looking at family Anolidae. And so what we're finding is evidence for this first hypothesis. We're finding a monophyletic Draconura, and then the Jamaican anoles as sister to this. These are really short branch lengths, so I know it could look like a polytomy. You'll have to just trust me there, it is not. So apletophalus and auratus are coming out as sister here to one another, and then our Jamaican anoles as sister to this group. So this black dot is indicating that we do find low support for this node. I think there's an obvious issue here in anoles of very low sampling of genomes. As I mentioned, there are over 400 species in this genus, and there are only 11 genomes present here. This is our current sampling. And then also we have low taxon sampling for within Draconura. So there is hypothesized to be over 150 species within Draconura, and I only have 2 representative lineages here. So this could be influencing this low support for this relationship as well. And so this has important biogeographic implications, the difference between these 2 different hypotheses of where we're placing Jamaican anoles in the phylogeny. And so this inferred sister relationship between a monophyletic Draconura and Jamaican anoles, tells us that the Jamaican radiation and Draconura would be the same age, and this is incredible. So within Jamaica, we have 7 species and over 150 in Draconura. Obviously, there are range differences here with, much more area in South America for these anoles to expand to, the Draconuran anoles. But the estimated age of Jamaica is 15 to 20 million years old, so this would be, incredible speciation rates within Draconura. So, obviously that comes with a bit of skepticism on if this is the correct relationship. The implications are kind of striking. So I think this definitely deserves a closer look, and I think we'll get that with more genome sampling. So that brings me to my conclusions and future directions. So we've assembled the first Jamaican anole genome assembly. This is a high-quality genome assembly that's allowed us to kind of dive into some interesting evolutionary questions that have been brought up by past research. We've identified the X chromosome in this species, which was previously unknown. And we see that it's a result of autosomal fusion with this ancient X chromosome. And then we also see significant clade differences within Norops as well. And then this, for the first time, we could investigate the placement of Jamaican anoles using genome scale data. Sorry, I'm hitting 11:45, so I'm going to really hurry up. The future directions, we obviously want more work on Jamaican anoles. Looking at sex chromosome evolution can tell us about, their process in the adaptive radiation, their role in the adaptive radiation of Jamaican anoles. And we, of course, want more genome assemblies from Jamaican anoles, as well as dibamia, if we're going to be looking at some of the squamate relationships that I didn't talk too much about here. Sorry, I am being conscious of time and hurrying. So with that, I want to thank my lab as well as my funding sources and, of course, all of the lizards that give their lives for us to do this really important research. I want to make sure they get their credit. And then a little plug, I'm part of the Open Consortium for Squamate Genomics, so if you are interested in doing squamate genome research and would like to join, there's a QR code here, or you could reach out to Brendan Pinto. And with that, I will stop talking.
All right, well, good morning, everyone, and I'm pleased to have this opportunity to tell you about some of the work that's being performed in our group that's primarily focused on the cnidarians. And this is a phylum that includes the corals, the sea anemones, the jellyfish, and the hydroids. And the distinguishing feature that unifies all of the members of this phylum is that they possess cnidocytes, and these are specialized stinging cells that they use to either ward off enemies or to capture prey. These cnidocytes contain a bulb-like capsule called a nematocyst, which, when triggered either in response to chemical cues or to mechanical cues, discharges a barbed thread with tremendous force. You can see the acceleration rate right there in a matter of nanoseconds, releasing toxins into its target. So truly a very impressive mechanism from a morphological standpoint and one of the fastest biomechanical processes in nature. From a genomic standpoint, they occupy an informative position on the animal tree as a sister group to the bilaterians, and from a biomedical standpoint, they encode more orthologs to genes associated with human disease than do classic invertebrate models, such as Drosophila and C. elegans. So this supports the proposition that these organisms can indeed serve as viable and models for studying various classes of human diseases, and we're going to use that as our jumping off point to talk about one particular cnidarian called Hydractinia, and these are colonial marine hydroids that grow on the surface of hermit crab shells. And what makes them interesting to us is that they're one of only a very small number of organisms that can regenerate their entire body throughout their lifetime. They've already proven to be excellent models for the study of regeneration, allorecognition, and stem cell biology, with most of the work focusing on one particular Hydractinia species that's native to this side of the Atlantic called Hydractinia symbiolongicarpus. So a little bit about the organism's morphology. Each clonal polyp, whether we're talking about a sexual polyp, which is the one on the right, or a feeding polyp, the one on the left, the other way around, left and right, is interconnected through a stolonal mat that's about 3 millimeters thick. And in addition to providing tissue continuity and a common nervous system, it has a gastrovascular space that's formed by a network of endodermal canals. More importantly, the circles that you see in the diagram are migratory stem cells. These are the circles again that are in the interstitial spaces of its epithelial cells. So because of where they're located, these stem cells are called interstitial cells, or eye cells for short. The most important thing to know about these eye cells is that they express genes that we know are involved in stem cell biology in bilaterian species. So the bottom line here is that it's these eye cells that provide Hydractinia with its lifelong capacity for self-renewal and are the basis for tissue regeneration. So let's see that in action. So we're going to just lop off the head of a polyp. After 3 hours, we see that it's closed the wound. By 24 hours, we have the formation of the blastema. So this is the mass of cells that's capable of proliferation and regeneration. At 48 hours, we start to see regeneration of the head structures, and by 72 hours, the polyp has completely regenerated its head. Fortunately, one does not have to depend on the hermit crabs because they grow quite easily on microscope slides in the lab, and as you can see from the list here, it's quite amenable to experimental manipulation. It's also amenable to single-cell methodologies, and we're going to talk about that in a little bit. Now, this is a genomics session. I think everybody appreciates that with any emerging research organism, the research can only go so far in the absence of high-quality whole genome sequence data. So given their potential for studying key questions in evo-devo and regeneration, cell biology and aging, we formed an international consortium to generate whole genome sequence data from Hydractinia. And this consortium includes Uri Frank at the National University of Ireland in Galway, Pauline Cartwright at the University of Kansas, Matt Nicotra at Pitt, and a special mention goes to my former postdoc, Christy Schnitzler, who's now an associate professor at the Whitney Marine Labs in St. Augustine. The genome paper was just published a few months ago in Genome Research, so rather than go through the usual set of assembly statistics in the short time that we have together, I'd like to instead take you through some very interesting vignettes that were made possible by having the quality of whole genome sequence data that we were able to generate. So let's start with some work that was done by Sylvia Barrera in my group, where she was exploring a possible correlation between the structure of the ribosomal repeat region and regenerative capacity. So in the top, we have the structure of the ribosomal repeat regions in human. So the usual structure that you see containing the 18S, 5.8S, and 28S genes, as well as the externally and internally transcribed spacers, a large intergenic spacer containing the gene and spacer promoters, the repetitive enhancer elements, and the transcription terminators. Now, when she compared this to what she saw in Hydractinia, she found something completely different. First, as you can see, there's many, many more repeats in the Hydractinia genome. Within each one of those repeats, everything is in the same order, but that 28S gene is over a kilobase shorter. You have an almost imperceptible 3' ETS and a lot more repeats with very short spacers in between, 100 times smaller than what you see in the human genome. So this is both puzzling and intriguing at the same time. Taking a step back, we think this might be an evolutionary adaptation that's necessary to meet the high demand for ribosomes and in turn protein production required during regeneration with that high copy number that you see, giving hydractinia the ability to just start cranking out cells and tissues in order for the organism to regrow. As I had alluded to before, we've already dipped our toe into the single cell pool, performing single cell analyses on suspensions of dissociated polyps. The number of cells were limited, partially because of the pandemic, partially because of experimental considerations. The methods that we were using at the time were not optimized for creatures that are in marine environments, and that's probably a drawback that many of you have faced as well. But regardless, we were able to identify 18 clusters that were then classified as putative cell types. And these included distinct clusters of nematocytes, the gray bar at the top, and 2 I-cell clusters, the yellow boxes below. Okay. We subsequently validated these marker genes by fish, so we have 2 examples here. So Peewee on the left for marking I cells and their progenitors, PCNA on the right, a known proliferation and broad stem cell marker in other animals, and both of them are expressed in the I cell band in the middle of the body column. Okay, so focusing just on the I cells and their progeny, we found that the I cells are defined primarily by genes that are shared with other animals rather than being lineage-specific genes. So that's the long brown bar in this diagram on the right, as compared to any of the other sets of cnidarian-specific genes in the row below. So we have the cell types going down the side, with the color bars reflect the degree of orthology to genes in the various groups that were examined, whether they were within the species, within the hydrozoans, within the cnidarians. 317 of the genes that mark I cells were present in the 43 animal species that we looked at as part of our orthology inference analyses. So this provided to us evidence that the toolkit employed by these cell types has a shared ancestry with other animals. So that's a finding that we feel can provide a foundation for understanding evolutionarily conserved functions of stem cells in other animals. So let's turn our attention to allorecognition, the ability to recognize self from non-self. And this is dependent on being able to generate immunological diversity. And one very conceptually simple way to generate diversity is to just have a large repertoire of immune receptors encoded within the germline DNA with high levels of allelic polymorphism. And that ultimately has important consequences for genotype survival. There's been a long-standing question as to whether invertebrate allorecognition genes are homologous to the vertebrate histocompatibility genes. And what we found is hydractinia does indeed have its own allorecognition system, but one that's much more complicated than we expected. So a little bit of background. These colonial invertebrates are capable of allorecognition purely on the basis of cell-cell contact as part of their competition for space in the environment. So here we're looking down from above. Sometimes you have contact between compatible colonies, the situation on the left, and that results in a fusion result. They'll just fuse together. But sometimes you don't, and this is where the nematocytes that I told you about earlier come back into the picture. During the rejection response, there's an extensive recruitment of nematocytes to the contact area, and the colonies just basically start shooting at one another. and so obviously causing extensive local damage to the adjacent colony. Now, for years it was felt that this was controlled by 2 genes called ALR1 and ALR2, allorecognition 1 and allorecognition 2. So if we look at what was known in these inbred lines, if you have a situation where you have 2 colonies that share an allele at both the ALR1 and ALR2 locus, that would result in fusion. There's an intermediate stage that it comprises a gradient of responses where if they share an allele at either ALR1 or ALR2, you get transitory fusion. So they come together, they kind of check each other out, they decide whether or not they want to fuse, but in most cases, they just sever their connection over a period ranging from hours to sometimes weeks. Finally, the situation where they don't share alleles at either locus results in rejection. So again, these are in the inbred lines. Of course, this is not what happens in nature when you take a closer look. So what ended up happening was when you look in these these other lines in nature, the situation where you would expect fusion because of sharing alleles at ALR1 and ALR2, sometimes you do indeed observe fusion, but sometimes you observe transitory fusion. In the situation where they share one allele at one of the genes, you'd expect transitory, but you observe rejection. So of course, all of this data suggested that there were additional allorecognition genes at play in the hydractinia genome. So now we've got a whole genome sequence. We went hunting, and we found that there were indeed many more, 41 ALR-like genes located in the allorecognition complex of this organism, spanning just shy of 12 megabases. Most of them are clustered together in one of three clusters in the gray boxes. All but two were in the arc. Okay, I would refer you to, given our time is short, I would refer you to Aidan Hune's paper published in PNAS for more information on the domain architecture of these ALR proteins and some very interesting structural studies using AlphaFold to identify hallmark immunoglobulin domains. So using these data as a foundation and looking through a longer evolutionary lens, we're currently looking into that question whether or not there is macrosynteny between the ARC and similar regions in other organisms, including the major histocompatibility complex, which in turn is thought to have arisen from a pre-vertebrate region called the primordial immune complex, with these ALR proteins showing structural similarity to a number of proteins that are encoded in MHC paralogous regions in humans. So this has led us to ask whether there is indeed a deep evolutionary connection between the allorecognition complex and the MHC, or whether it's a case of convergent evolution. So Alberto Rivera, one of the postdocs in our group, is actively working on this question, putting AlphaFold through its paces to look for relevant structural similarities between cnidarian and early vertebrates proteins involved in the immune response, and you can learn more about his work at the molecular evolution session tomorrow morning down the hall. Okay. So with that, I'll leave you with a picture of some of our collaborators during our biennial NidoFest meetings, the URL for the website describing the hydractinia genome in more detail, including a genome browser and a single-cell browser, so you can go through our single-cell data, and most importantly, the folks in my group that I have the pleasure of working with over the years. So my thanks for your attention, and I'm happy to take one question to keep us on time. Thanks for your attention.
Okay. Hi, everyone. My name is Walter, and I'm a postdoc at the University of Florida within the Blackburn Lab. And today I will share with you some of the recent results of what I've been doing down there for the last 2 years. And this is a collaborative work with my co-postdoc, Ethan Allen, and several undergraduates that I've helped to process all this huge amount of data. And thanks to my PI, Dr. Blackburn, for hosting us for this long. Okay. A little bit of background on African biodiversity. So tropical Africa has undergone a long and complex evolution, resulting in a spectacular and unique biodiversity. Several authors consider the Congo Basin as the core of the African rainforest. So the Congo Basin, as you usually know, extends from the Atlantic Ocean in the west towards the Albertine Rift Mountain in the east. So this forest block is highly similar to the Amazon forest in South America, and is ranked as the second largest rainforest in the world. So within the Congo Basin, past work have aimed at understanding the phylogeography, speciation, and adaptation mechanism of living organisms with high dispersal abilities. So the past events such as the climatic oscillation and the formation of geological fluid structure are known to be some of the causes, such as the common volcanic line on the west, Central Africa, and the Albertine Rift on the east. So two main scenarios have arisen, suggesting that from the climatic oscillation, which are the interglacial refugia or the evergreen rainforest. Okay, let's now dive into the group that we're talking today on. So there's 4 families of neuron that are known to be endemic strictly to continental Africa. The clades is called Afrobatrachia and are diverse nearly 90 million years ago. So it's composed of 4 families: the Archaeopterygidae, Hyperolidae, Brevicipitidae, and Emisotidae. So the group that our working group is found is the Archaeopterydae family, which is the second most species, widespread in sub-Saharan Africa, and it's made of 3 subfamilies, and the one that we'll be working on is the subfamily Acylostaninae. So within this subfamily, with the exception of 2 species that belong to the genus Acylostanus, the entire remnant member of this clade are found to be strictly endemic to Central Africa. Okay, the group itself. So the night frog is composed of 2 genera. We have the Acylostanus genus, which is composed of 12 species, and then we have the Trichobatrachus, also called the hairy frog, which is a monotypic genus. They distributed mostly in Western South Africa, as I said earlier, and to date we have those two main clade, which are still the issue here. And they are endemic to rainforest, so they are really associated with that type of habitat. And they have this unusual biology. They are really like the only way to find them or place and spot to find them is in fast-running, super clean water within the forest. They are known to have retractable claws on their hind limb, which have given them the name of the Wolverine frog, because, yeah, you will bleed. That's my finger over there. And also in the group known as Trichobatrachus, the one that is usually called the charismatic Wolverine hairy frog, the male, when they are during the breeding season, they grow hair-like structure and they present reduced lungs on the inside. And more interesting, guess what? We found one species there being polyploid. So due to this polyploidy, so several researchers, when they have attempted to do nuclear sequences on this group, ended up having a high number of heterozygosity. So they first think, oh, this is just garbage. Maybe the sequencing didn't work, and they ended up discarding a lot of data. So if you work on frogs and you find high heterozygosity, don't throw it away. The other thing is the phylogeny is not well resolved because people, due to the fact that they cannot use nuclear data, they only use a limited amount of mitochondrial data, and which led to an unresolved phylogeny in this group. And also, no one has attempted to look into the diversity and phylogeny relationship within Acylostanus and or Trichobatrachus. So in this talk, where I stand right now in my research, we'll try to assess the phylogenetic relationship of Acylostanus Trichobatrachus using mitochondrial genome. We'll try to assess the phylogeographic impact of the camovolcanic line in the Central African clade, where most of the diversity of this group is found. And finally, we'll determine the prevalence and the level of ploidy across the entire clade. To achieve this, we generated frog cap data from 325 samples spanning the entire 13 species known, and we have one of the largest sampling within the group. We used this pipeline designed by Carl Hutter, frog cap, to generate several loci and markers, and we use this mitotrol-R pipeline to extract mitochondrial genome because we're still processing the data as of now. Like this morning, I was running some analysis still. And we also inferred phylogenetic relationship tree with the mitochondrial genome, and we use ploidy NGS to try to infer the ploidy level. Okay, so at first we have the first level phylogeny, which kind of give us a well-resolved and well-supported branching of the entire clade. But this time, Trichobatrachus pops out as sister to the entire Acylostanus clade, but still, the West African clade is still not properly resolved in its position. And also, we find a high level of diversity within the Central African clade, which there's probably some more look into it. Okay, so another thing that pops out when we did plot all of those individual, there was a high number of misidentified species within the clade, which can happen because most of these species occur sympatrically in different areas. So if we start with the West African clade, it's splitted from the Central African clade by the Dahomey Gap, which is this area here east of Ghana. And then within the West African clade, there's also a second split, which generated those 2 species down there. And the separation between the 2 species is mainly the currently disappearing remnant Upper Guinea rainforest in the southern Ivory Coast. Okay, so let's look now into the high elevation one, where most of the diversity in this group is found. On the high top there, that's the Cameroon volcanic line. It's a mountain channel that goes from the Atlantic Ocean in the island of Bioko and goes towards Cameroon, enters Nigeria, and comes back in the country. And up there is a profile showing you most of the mountain and some kind of the different level elevation that is spanning the distribution of these frogs. Two species are, and usually, let's back to it. The entire Cameroon volcanic line is split into two major spots. We have the southern Cameroon volcanic line that has more of like a rainforest, southern Atlantic climate. And then we have the northern part of it, which has two more types because the more it goes north, the more it goes toward the desertic area. So it gets more arid, and we have some kind of transition area in the area called the Bamenda Highland. So two species, parity, and montanus are endemic to the southern part, but still show some phylogenetic structure within the trees. And then we have these three other groups which have a distribution that is spanning all across both the southern part and the northern part. And all the interesting thing is the position of the species Asylocynus ganghanus, which since its description in around 55 years ago, was never plotted into any phylogeny. So it ends up, it's a strictly endemic to the northern Cameroon volcanic line, and it's at the end of the tip of the entire Cameroon volcanic line in the Adamawa Plateau. Okay, so let's now go back into some more low elevation species. We have the Laurenti phylax paraphyletic clade, which span across the southern part of the Cameroon volcanic line in like low to mid elevation area, and it's more likely to be three different species which require more attention. And then we have the most widespread group, which is the clade Battersea schutzi. Schutzi is a very cryptic species. I don't know if you can see the star up there in the maroon circle around. The star is the holotype area, and the circle is the only sample that we have in the soil material. And then we have the Battersea clay that span all across southern Cameroon all the way to southern Gabon. It's pretty widespread species, also very associated with clear water and rainforest. Okay, since this is a genomic session, we were able to process all the data for only one group, the hairy frogs. So here's some a cut down through what we were able to generate using FrogCap. The program targets nearly 34,000 markers, but we were able to amplify 13,000, which we generated a concatenated read for worth 17 million base pair. The shortest marker was around 119 base pair, and the longest was near, a little over 50,000 base pairs. So we did a concatenated phylogeny using IQ3 on these, and we compared here versus the mitogenome. So both of them uncovers three major population, a lowland one, which spans from, which is the blue one here, spans from Cameroon all the way to southern to northern Angola, which is incredible. And then we have an endemic population that's found only in one mountain at an elevation like above 900 meters. And then we have another one that's in black, which is endemic to the southern part of the Cameroon volcanic line. The mitogenome and the FROCA were pretty well resolved, with the exception, of course, of a few mitonuclear discordances. What I want to point out here is the light blue spot on the outside branch there. So we found one Trichobatrachus that the nuclear material is not monophyletic with the entire order Trichobatrachus. Guess what? There'll be a surprise later on that. Okay. Oh, I don't know what happened here. Oh, my slides disappeared. Anyway, so when you use Ploidy NGS, I'll try to say and be graphic, and hopefully it makes sense. So when you use Ploidy NGS, it generates, you can have apply a filtering to it, and it goes through the BAM file. And if you have a diploid species, you get all your sample presenting just one peak in the middle. If you have a tetraploid species, you get 3 peaks, one at 25, one at 50, and another one at 75. So one is a topoploidy, you get 3 peaks of almost equal height. But when you have a case of alloploidy, you get 3 smaller peaks on the outside and 1 giant peak in the middle. So we use Astroleptis porcinatus, which is a non-diploid, and normally should have a deep peak in the middle. And then for Diadematus, which is the known Acylostanus, it has 3 peaks. Sorry, it's not showing here. We did apply the same program to our entire data set. Thank God this one, the others, at least the tip are working. The black tips are the ones that were found to be diploid. The red ones are the ones that are found to be tetraploid. And the blue are the one where we had just a teeny tiny bump. So it's not as flat as what you expect from the diploid, and it's not as high as you expect from the tetraploid. So ends up, we have way more polyploid frogs in the Convolcanic as previously known. And if you remember the blue line within Trichobatrachus, is that guy. It's a sole one full tetraploid that we found within our entire Trichobatrachus data set. So, which is pretty cool and kind of scares me a little bit because there's more stress coming with that. So what is the takeaway from it? So the genus Acylostanus joins a growing list of polyploid Central African frogs. Xenopus, which is an interesting other one, which it goes from tetraploid to dodecaploid. We have Opelbatrachus, which is a tetraploid species. The Convolcanic 9, as you can see in this paper from David in 2022, is encompassing an area where there's some kind of presence of polyploid species. We expect here as a hypothesis that the climatic oscillation and during the Miocene and Pleistocene has led to niche expansion and contraction, which has brought different species to disconnect and come back in contact and make hybrid to what polyploid in this case. If you want to hear more about polyploid frog, there will be a talk given by my collaborator, my postdoctoral Kate Allen, in the room 516. It will be at 10:15 tomorrow, and I will give a thank to all my fundings and collaborators, and I'll be happy to take any question if there's any. Thank you.